Hello students, hope you are doing well. Today is the last part from lesson 11. We are going to cover the last pages 287 to 292. So the topic that we are going to focus on is um, condensation polymerization. So one big example for condensation polymerization is a nylon. Nylon is a good choice to use the condensation reaction for making polymer of nylon. So nylon is a copolymer of two different monomer. You can see at the right side that there are two different monomers. One is diamine, which there is two amine group at the terminal. Okay, so one at the left and the one at the right. You can see diamine. Diamine has amine group. If any compound has two amine group, then called diamine. Okay, di here meaning means two, and another second monomer is a carboxylic acid. So carboxylic acid means a compound that has carboxylic acid group. That means COOH. If any compound has this chemical group, that means it has it is acidic, and the functional group, the chemical group is called carboxylic group. So if you look at the picture at the right side, you can see this functional group HOCO, and this is carboxylic group. So the reaction is happen between an amine group in one side of the first monomer, and uh, with another carboxylic group from the second monomer, another group. So the important part is sharing, not sharing actually, it's removing one hydrogen from amine group and one OH, uh, hydroxyl group from the another monomer from carboxylic group. So you can see that these two parts can be removed, and then they will form they will form a water. So water is um, um, is one product. One product is water, which can be removed from the main reaction, and the main product is a nylon. Nylon is a polymer. You can see there is a connection between amine group and the carboxylic group. So between these two group, uh, the bond is called amide link. Okay, here you can see that um, the type of bond between amine group and carboxylic group is called amide link. So that's why this type of polymer, also well known as polyamide, is called polyamide also. This is a condensation polymerization, it's called. Then in this slide you can see after reaction occurred, then one rod can be rotated and collect the nylon in the solution of water. So nylon fiber is pulled out by rolling the rod or any stick. So all right. And another example for condensation polymerization. Okay, condensation polymerization. First is nylon. Please concentrate on this reaction. It's very important. Keep in your mind that amine group one side and another is carboxylic group. And second example based on your book is polyesters. Polyesters are the condensation polymerization made from two monomer like the previous one, but one side is alcohol group, not amine group, and another side is carboxylic acid group like before. And you can see on the right side here any compounds that has two side, two terminals with the alcohol group or hydroxyl group, also both are same. And another one same as before, another monomer should contain a carboxylic group at the terminal two sides. Then one hydrogen from alcohol group and one hydrogen and oxygen from the acid group, carboxylic acid group, can react together and then produce a water, can be separated as a form of water, and then the rest, the main chain of the chemicals, can produce the polymer, which is called polyesters. This type of bond between one oxygen from what we call from the alcohol and the carbon from another monomer by carboxylic group. So this type of link is ester link between oxygen. You can see between oxygen and carbon, which carbon has a carbonyl group. So this type of chemical group is called ester. The type of bond or the link is ester link. So that's why this polymer is well known as polyesters also. All right. Um, you learned addition polymerization in the previous part. Now you have learned condensation polymerization. So there are some similarity and there are some differences also. But this table is very important to compare addition polymerization and condensation polymerization. Please have a look and memorize this table. So in the time when in the exam, if you will be asked about some condensation polymerization or addition polymerization, then you will know about comparison or differences or similarities. All right. And the next part is the reuse, recycling or disposal of plastic waste. We have learned about nylon. Nylon is a plastic, so it is very important to recycle because it is very dangerous for environment. Um, you've seen that um, there is a connection between the monomers, so that monomers will not be easily to be separated again. So it's a polymer, all monomers are connected to each other, so they will not be broken down easily. So this part is for you. Just have a self-study on this part, and let's move to the next part, which is biological polymer. So we have biological polymers also that are not made by human artificially. Naturally they are available. Also nowadays scientists are able to artificially make biological polymer like protein. Protein is one of the common example for biological polymer. Proteins are what cells are made of. So our cells in body, or any organism's body, are able to produce protein. Proteins are very important in order to construct some part of the organisms, and also it's very important for any function, any metabolism that happen in the organisms. living things. Like for example, DNA in our body that are responsible for transferring information from parent to children. So DNA also can be preserved, can be protected by proteins. There are many proteins in DNA to protect the form of protein, and many other functionality in our body are rely on proteins. Proteins are built from amino acids, so each monomer, actually we learn about monomer and polymer. So if we consider that protein is polymer, the monomers are the unit cell to construct protein is called amino acid. So amino acid in protein play a role as a monomer. And there are totally 20 different amino acids are used for constructing a protein, but the arrangement of these different 20 amino acids are the type being used for making proteins are important, so they will produce a wide variety of protein. So another glycine and alanine are two of the simplest amino acids. So I said that we have 20 different. These two are the simplest one, and so if these two join together, then they make a dipeptide. Dipeptide is a peptide. It's not protein, because protein is a bigger size, but peptide is a small size of the protein. If only two amino acids join and make a peptide, actually, so it's called dipeptide. Protein is a larger size. Peptide is only a few, like two, three, four, five. Small number of is including a small number of amino acid. So at the right side you can see the structure of the glycine and aniline. That aniline has one side amine group, nitrogen and two hydrogen, and glycine has a carboxylic group in one side. So this carboxylic group, the OH or hydroxyl group of the carboxylic group, can react with the hydrogen from amine group to form a water molecule, and then the rest is a dipeptide. So please memorize for this reaction structure, also keep in your mind. So the type of link between these two monomer is an amide link. Okay. All right, how we can break down on protein. Proteins or other polymer also are able to be broken down. They are able to be um, uh, hydrolyzed. So one one way that they can be broken down is uh, hydrolysis. Hydrolysis, that means using water. We have another way, that is thermolysis, if I'm not mistaken. That is only using only temperature, high temperature. But hydrolysis is using a water, sometimes in high temperature. So if we add water to the what we call the protein that already been synthesized, the polymer or protein or any kind of polymer. Polymer can be biological polymer or industrial polymer like nylon. So any polymer can be broken down by adding water. Adding water. So if we add water is called hydrolysis. Hydro means water, okay? Hydrolysis, then you can break down a polymer to the monomers. okay, the units. But if if you extract water, okay, if you take out water from the reaction between monomers, that is called condensation. Okay, so please keep in your mind, for the hydrolysis and condensation, they are opposite word. And after hydrolysis, once we will get two different types of monomer, then they can be separated by chromatography. That is in another important term that you need to know. A mixture of amino acids can be separated by chromatography. This is one laboratory tools. So uh, we learned many different methods for separating phase, like gas, mixture of gas, or mixture of solid and liquid. But here you should know that mixture of two amino acid or other monomers also. Other monomers can be separated by chromatography. And another biological polymers here, we had one example as proteins as biological polymers, and here carbohydrates is another example for biological polymers. They are polymers like sugar. Sugar is a kind of carbohydrate, and the glucose is the simplest, is one of the common sugar, is used in fruit, in any sweet food. um, so the chemical formula is C6H12 and O6. And a carbohydrate is a compound containing carbon, hydrogen, and oxygen only. So these are very similar with the hydrocarbon. Hydrocarbons we learn in chapter 10. So this is carbohydrates, that only carbon, hydrogen, and oxygen. All right, and all long chain carbohydrate is polysaccharide. So long chain means polymer. Polymer form of uh, carbohydrate is polysaccharides. They use suffix poly for saccharides. That means a polymer is a kind of polymer. You can see the right side that two the two terminals in monomer for sugar is OH, or hydroxyl group. Hydroxyl group one OH from one monomer can join to one hydrogen from another hydroxyl group to form a water. So the rest after the reaction product, or any polysaccharide, will have oxygen. The link between the monomers is oxygen. So just for your information, this is ether. Ether is called ether link. The type of link is ether. That means two sides are connected to each other by one oxygen. Okay. Polysaccharides can be broken down in laboratory by warming hydrochloric acid, which is called acid hydrolysis. So also same as the previous one, like protein, here we can use hydrochloric acid, but having a high heater, using a heater to warm up the reaction. Then we can break down the polymer. And after breaking down the polymers, then chromatography can be used separate to separate the monomers. So that's all for this part. This is the last part of chapter 11. So please memorize and finalize for this chapter. Thank you so much. Take care.